I am a scientist turned entrepreneur, and I haven't been to the lab in the last couple of decades. I have no data to present. I'm only here to share what science can do. Am I audible?
Yeah, fine.
Several years ago, my son was a class 7th student. He came from a school equipped from his library, and he asked that, Papa, do you know what is the origin of bless you when somebody is sneezing? I said, no. He said, actually, in ancient times, when somebody was sneezing, they knew that the guy has some infection and that nothing can be done. So all they did was bless you. We have moved forward quite a bit. We have not only we have drugs, we have designer drugs. Today we can design a drug that we can command to perform a certain function. One such example is the development of a very novel liposomal amohotericin B in saline. Most of you sitting here who have ever had interest in fungal infections or studied antifungal drugs would probably know amohotericin B. The origin of this drug is in DBT. DBT had a mission program under which they wanted to bring self-reliance to India. If you do not know, the first liposomal amohotericin B was developed through a combination of teams from United States and UK. And the first product which was made is Invasive. And Invasive used to cost $240 per injection. And every day you needed 3 to 5 injections, about $1,200 per day. And at least for 21 days daily dose, sometimes for a month, and sometimes even 6 months daily dose, which was beyond the ability of an average Indian to be able to afford. So the DBT took a mission program. They started the development of this drug, and I am a liposome technologist. And when I returned from my postdocing from the United States, I took over this charge of taking it to the people, from laboratory to the hospitals, from laboratory to the patients. Interesting developments happened in last, I would say, 10 years or so. We launched the drug in 2003. And there are very few antifungal drugs. The interesting thing about fungal infection is that if it is a systemic fungal infection and the patient is not treated with the appropriate drug in time, mortality is 100%. So therefore, not only you need to start the treatment in time, you have to have an effective drug. There are 3 groups of compounds, antifungal predominant compounds, azoles, echinocandins, and amohotericin B. All azoles and all echinocandins have serious problem of drug resistance. All have the problem of toxicity, major toxicity, because fungal cell is a eukaryote and so is a human cell. So if there is a compound that works on fungus, in all possibilities, it also works on the human cells and causes significant toxicity. In addition to that, they all have limited spectrum. So the only drug which is broad spectrum is amohotericin B. It's very potent. So if you have a very potent drug, and if you have a broad spectrum drug, then what is the problem? The problem is it is a very toxic drug. Because it is very potent, so it has got to be very toxic drug also. It causes nephrotoxicity in nearly 2/3 of the patients who use this drug. Things are going all right. This liposomal amohotericin B in saline was developed here in India. The important point about this is amohotericin B is known to precipitate in saline. Therefore, of the very limited formulations that are there in the market around the world, there are precisely 3 formulations which are in the market. And they all write, do not use saline. We are the only one in the world who says, use only saline. we stabilized the drug liposomal amphotericin B. The liposome is also very strategically designed. So we made the drug in saline, which brought down the nephrotoxicity to zero. And the efficacy is double of that of the European drug, which is Invasive. In 2018, there was a major report in Times of India that there is a fungal infection called Candida auris. It is multi-drug resistant Candida auris and does not respond even to amphotericin B. I am making a very categorical remark here that this amphotericin B resistance is a mistaken conclusion that Candida auris is amphotericin B resistant. Some amphotericin B formulations, actually most amphotericin B formulations, do not work on Candida auris. But this liposomal amphotericin B in saline does work. And in a very limited time, I will give you that evidence. Since this is the only drug that works on Candida auris, on proven strains of Candida auris, and there's only one publication, I brought some copies of the publication to share with those who are interested in going through it. So in 2018, when this major report came, incidentally, I had a friend who was a microbiologist and he was visiting from United States. So I asked him to take up this study because he had access to the Center for Disease Control, identified the strains of clinical isolates of Candida auris, which are MDR, proven MDR. So my talk will not be dependent on these slides. I do have slides, but so this only shows, if you see the top line, the amphotericin B is the only drug which is effective on all major fungal pathogens. And I'm written here major, but actually it works on all fungal pathogens. You can see all of them have toxicity at the last column, and amphotericin B has major problem on nephrotoxicity. All right, you need not go through the finer details of this one. So you can see that the only drug which is Fungisome that has no nephrotoxicity. Okay. You can see there's a lot of drug resistance. Actually, almost all the echinocandins and azoles will have drug resistance problem. Amphotericin B will never have drug resistance. Never it will have it because of the special unique mechanism of action that it has. All right. So when I said that it is mistaken MDR, here is a report. When voriconazole was launched, the voriconazole was compared with the conventional amphotericin B, which is non-liposomal amphotericin B, and it was shown that it is not as good as voriconazole. It was a faulty experimental design only to prove that voriconazole is a superior drug. It was an industrially driven publication. Okay. This work was then criticized. World over, it was criticized that do not publish such misleading reports, okay, because you designed it to show that amphotericin B doesn't work. Similarly, in the recent years, since echinocandins have come to the market, there is an increasing tendency to show that because amphotericin B is nephrotoxic, so do not use that, use echinocandin. The fact is that in a major study which was done at the Harvard Medical School, they have shown that echinocandins have very limited antifungal activity, which is about 33% or so. Okay. Now coming to Candida auris. The first patient of Candida auris was identified in 2009 in Japan. When they saw this first patient, they went to some clinical samples which were stored from 1996 onwards and concluded that the Candida auris was there all around since 1996 and maybe even before that, but they identified it now. And beginning in 2009, now the Candida auris is everywhere in the world. Among the Candida infections, this is one of the major infections that is feared to be not curable. There are various reports about Candida auris. It's a major global problem nowadays. When I said that in 2018, I proposed that you undertake a study, it was based on earlier observations. When we developed Fungizome, and although it was clinically available, we still wanted to, we continue to do research and study. We are still working on the same drug. We gave one study to PGI Chandigarh. And there is an Indian expert called Arunabh Chakravarti. He is a professor at PGI Chandigarh. And they have a WHO reference laboratory. And so we gave for comparative MIC. And there he concluded that two observations, important observations. One, that the Fungizome, the product in Saline, is 2 to 16 times more effective than the conventional Amohotericin B, MIC. And the second thing that he said that this Fungizome works on Candida humulini, which was believed to be not sensitive to Amohotericin B. So because of that, we had a clue that this product is different, and any time you have a problem, you should check up with this one. 3 laboratories have studied so far, and they have concluded that the Fungizome is 2 to 32 times more effective in terms of MIC as compared to another liposomal Amohotericin B, which the word known as a Bizome. This was a study done by Chakravarti's lab, which I was referring to. This is a laboratory somewhere in Himachal Pradesh. They also said it is very effective. This is a study which has which has shown that it is 8 to 32 folds more effective than Bizome. And these are the various Candida auris strains from 5 different countries, and all of them have been shown to be sensitive to liposomal Amohotericin B in Saline and non-sensitive to other liposomal Amohotericin B. During Corona, there was a report that there were 2 patients in All India Institute of Medical Sciences, and they were the patients of Aspergillus lentulus. And both patients died because no drug worked. So we thought that we should also look at Aspergillus, and we tried to get hold of Aspergillus lentulus. To this date, we did not get that clinical isolate, but we undertook a study on the Aspergillus drug resistance, or you can say sensitivity to our product versus other products. And so far, the report says that all the Aspergillus tested so far that are supposed to be drug resistant, they are sensitive to Fungizome, and they are not sensitive to Bizome. This is a comparison which shows that the Fungizome has very high efficacy and very nephrosafe. It does not have any nephrotoxicity, in fact. And therefore, from these observations and other observations from the literature, we say that there is no Amohotericin B resistance in Candida auris. The only thing is that you have to test with the appropriate formulation of Amohotericin B. Okay, the same organism which is sensitive to Fungizome is not sensitive to Bizome. Therefore, you must do the sensitivity testing properly with all the formulations that are available in the market. And in addition to that, there are other results. We have seen this product in leishmanial infection also. In Leishmania, there was a problem very briefly that those patients which were treated with Invasive, left, right, and center in India, many of them, about 20% of them have developed PKDL, post-Kala-azar dermal leishmaniasis. We conducted a 7-year follow-up study. And in 5-year follow-up, all the patients who were treated with fungizome, we did not have one incidence of PKDL. Okay. So this is all that I had to say. Therefore, when we are working on the infectious diseases and we are looking for optimization of the drugs, we must have the carrier designed very carefully in all possibilities. If you can design your carrier well, you will get good results. Thank you very much for your patience.
Long live India and France friendship.